Hello students, welcome back to the next module of this chapter. Now today in this module we would be learning a very interesting process, that is the formation of urine. And my friends, formation of urine is divided in various stages, and the first stage we are going to study here is called as ultrafiltration. But my friends, ultrafiltration is quite a huge process, so I have divided this into two parts, that is ultrafiltration part 1 and ultrafiltration part 2. Now let's begin with the formation of urine, and this is your beautiful diagram of nephron, which is the structural and functional unit of kidney, where what occurs? Yes, formation of urine occurs inside the nephron, and this process is divided in three stages. The first stage is called as ultrafiltration, the second stage is called as selective reabsorption, and the third stage is called as tubular secretion. Now, the filtrate which is obtained after the first stage, that is ultrafiltration, is called as primary urine. And my friends, that is not the final urine, because it will later on undergo the next two stages, and after tubular secretion, the product what is obtained is called as your secondary urine or the final urine, which is ready to get expelled outside your body. Now let's begin with the first stage, that is ultrafiltration. And once again, one more beautiful diagram here, that is of Malpighian corpuscle. And as you can notice the name, that is ultrafiltration. What is ultrafiltration, my friends? Yes, the main key word here is filtration, filtration of the blood. And this filtration of blood is occurring under pressure. And what is the structure here present? The structure is called as the Malpighian corpuscle. And since this process is occurring under pressure, this process is a physical process. So ultrafiltration is a physical process. Now there are various other factors also responsible for this process. Now let's see these factors one by one. Now let's see the first factor here. If you notice this diagram, once again this is a diagram of Malpighian corpuscle. If you notice, a very important thing here is that if you notice the difference of the diameter of the arteriole, the incoming arteriole which we already studied, which was called as afferent arteriole. Notice the diameter. The diameter is quite large, and if you notice the diameter of the efferent arteriole up there, it's quite narrow. Now what will occur due to this? Due to this difference of diameter, where afferent arteriole has a huge diameter and the efferent having a narrow diameter, what will happen? The blood will come at a faster rate than it leaves it. So what will occur? There would be huge pressure created inside the glomerulus, and this pressure is termed as hydrostatic pressure. So the pressure created inside the glomerulus is termed as hydrostatic pressure, where this word is divided in two parts, that is hydro and static, where hydro means water, that is the water of the blood. And since it is constantly coming in, and what is happening? The blood is leaving at a very slower rate. So there is accumulation, there is stagnation of blood inside, due to which there is a pressure created. And that pressure is of the water itself. So that's why it is a hydrostatic pressure, with the constant stagnation of water inside. Now let's see the another factor. The another factor is due to the capillary of the glomerulus. So if you notice, or you can see the capillary here, which is highly porous, you can see a lot of pores over it. So what will happen? There would be a filtration at a higher or faster rate. And if you notice, my friends, this capillary is a specialized capillary only seen in glomerulus, and it is 10 to 1000 more permeable than all the other blood capillaries present in your body, due to which what will happen? Lot of substance will get filtered out. Okay, but not all the substance, only low molecular weight substance and water. Hence, since it is only allowing low molecular weight substance and water, that's why it is a semi-permeable capillary. And it will disallow larger molecular substance such as plasma proteins would not exit from these pores, as well as your blood cells, and the larger fat cells are not allowed from these pores. So this is one of the factor of ultrafiltration. Now let's see the another factor responsible for ultrafiltration, and that is the Bowman's capsule. If you notice, my friends, Bowman's capsule looks like a cup. So it's a double-walled cup-like structure where the outer lining is formed by the outer parietal layer, which is made up of squamous epithelium. And the inner lining of the cup is formed by the inner visceral layer, which is also made up of squamous epithelium. But here, the squamous epithelial cells are modified into a specialized cells which are termed as podocytes. Now let's see what are podocytes. If you notice here in this diagram, the yellow colored cells, the huge cells which are embracing the blood capillaries are called as podocytes. Now let's take out one capillary and see the podocytes. If you see this structure here, there is a blood capillary over which there is a podocyte embracing it. So what do you mean by podocyte? Well, podo means the feet-like processes and cytes meaning cell. So this feet-like processes, the gap in between the feet-like processes is called as the filtration slits. So this filtration slits also makes the ultrafiltration excellent. Okay, so this was the podocytes. Now due to this, due to the high pressure of the blood in the glomerulus and due to the porous nature of the capillaries, the substances are forced out, and due to which about 1/5 of the volume of the blood is filtered at a given point of time. So this was all about ultrafiltration. So we studied formation of urine, ultrafiltration part 1. In the next module, once again, we will continue with ultrafiltration part 2. Till that time, take care. Thank you.